Well, thank you very much, Mr. Chairman. First of all, I would like to thank the University Gajah Mada and the Faculty of Medicine and all the organizers for inviting me to come to this symposium. And as you will see later on during the presentation, we do have, and I personally have, collaboration going on with scientists from Gajah Mada. So it's always a pleasure for me to come back here. So the more I can come, the more I'm happy. So, to continue on this, when I've been invited to give this talk, I've been invited first to give a talk on this title, on the current concept of dengue vector control. And my first reaction was to say, well, there is nothing new, really. And then second thought, immediately I said, well, it's a good idea, actually, because there is a real need of getting together around the first word of the title, the second word of the title, I mean, the concept. It's very important to reconsider all the concepts, and think about what we are doing, what we want to do. And that's what I'm going to do in this presentation, is mostly to navigate around the different concepts related to the vector control itself. Sorry. Okay, so the first question we should ask ourselves, okay, we're talking about vector control. Okay, but what are we talking about? What is it? What is it really? Vector control. What do we want to do, and what are we addressing? And if I try to summarize that in one single word, I would say we are dealing with dynamic. We are dealing with something which is moving a lot, and so we have to really consider that. So if we do some flashback and we consider the origin of dengue, something that has been addressed already yesterday. Dengue is a disease which is a selvatic origin. It originates from Southeast Asia and Indonesia, actually. And it's found initially on non-human primate. So it's a zoonosis where 4 different viruses became to infect humans. Okay, so it's a vector-borne zoonosis with a major vector, which is Aedes aegypti. That is the origin. Let's go a little bit further. We have another dynamic, not only from one species to another, but also now we have a kind of geographic dynamic. From the beginning, this disease is expanding. Okay, first in Southeast Asia, where it originates, later on in Latin America, currently in Africa, Europe, and most likely North America are next online. And so it's really expanding, another kind of dynamic we have to think about. This is an additional factor for the dynamic. It's the globalization. We are living in the global world. Everybody is exchanging with everybody. A lot of movement, movement of population, human, a lot of movement of goods also. So this is very good for spreading the mosquitoes around. Okay, and we do have another element of the dynamic getting into the picture. It's another vector, Aedes albopictus, which is not the historical vector of dengue, but it's also vectoring dengue, and it's a mosquito that adapts very well to other places. So another element we will consider. So, as I said at the beginning, for dynamic, in my opinion, is really the key element. And this from the very, very origin, because we know the virus originates in Southeast Asia. Fine. 4 independent emergences which occurred locally, no problem. However, the first problem is that the main vector is African, is not Southeast Asian. And we do have a secondary vector this time, which is Asian. And this secondary vector, which is Asian, is undergoing some sort of worldwide development, colonizing many places all over the world. So from the very beginning, we had already some specific aspect of dynamic we have to consider with vector control. Because we focus on vector control, on the vector, but it's just a consequence. The origin of everything is actually the human activity. Is the role of humans. Humans are responsible for everything at the beginning. Aedes aegypti was brought from Africa to Asia through business. and through the intercontinental trade, the exploration, and for spices, business for spices, wood, slaves also with the American continent. And it was also the time of colonization and large seaport settlements. And actually, when you go back to the old literature, dengue was very initially described as not as dengue, but as a seaport and river disease. It was affecting the people living in the seaport or on the river close to the port. And initially, also we have vectors, mosquito vectors of dengue in the Sylvatic cycle, which are not anthropophilic. They are not initially prone to go to the human habitat. But Aedes aegypti is anthropophilic. And it's also the first consequence, the first dynamic, is that because of human activity, we brought a vector which is not the native vector of dengue, but is a mosquito which lives very well next to humans. We brought this mosquito to a place where dengue was present, and dengue jumped from their natural vector to Aedes aegypti. And of course, the things went very well because this vector was a good vector and anthropophilic. So he is a good vector because he can, of course, accept the virus and transmit the virus. He's also a very good vector because he lives next to the human and likes it. So, and it's very important to consider it's not the natural vector initially, from the biological standpoint. It's a secondary one. Just to say that in the future this could happen. We could have a jump to another kind of mosquito in another region. Why not? It happens once, even twice with Albopictus. Why not another time? So it's we brought the vector. So we keep focusing on the vector control, etc. And I think there is a very good story about the dengue and the vectors and the focus on the vector, because we think of vector control. Vector control means we have to eliminate. Usually, vector control translates into vector eradication, vector elimination. Which might be a very big mistake, actually. And the story of South American case of dengue is a good example of that, because the historical problem in Central America is not dengue, is yellow fever. And even one reason why the French stopped it's not the only one, but the good reason why the French stopped the construction of the Panama Canal, because many people were dying of yellow fever. Then insecticides came, the mosquito was eliminated, and there was a large campaign of eradication in South America, which worked very well, extremely well. And the mosquito was indeed eradicated, and yellow fever was indeed largely eliminated and controlled. Well, the vaccine came after that and helped a lot, but the elimination of mosquito was a good success in terms of eradication. However, what happened? When you leave an empty space, nature hates that. Nature is going to fill it up again. Okay? So that's exactly what happened, and new populations of Aegypti came, this time from Asia through, trades, and they came to Asia, they occupied the empty ecological niche, and they brought along dengue. So we just replaced yellow fever by dengue. Okay, and dengue is now expanding a lot in Latin America. Same thing, more or less, is going on at the moment in Africa, where dengue is expanding. And what about Europe? Okay, people in Europe usually think, "Woof, it's not a tropical country. It's too cold, no problem, no big deal." Dengue is other people's disease. It's a tourist disease. We don't have to worry about that. Well, not that much, because we have to run things to consider: climate change, Aedes albopictus, which is more resistant to lower temperature than Aedes aegypti, and is also a good vector of dengue or chikungunya, for instance. And this mosquito has already starting to colonize Europe. And it's coming from Southeast Asia, Through what? The very intensive commercial exchanges going on between Asia, Southeast Asia, and the rest of the world. So we are still in the same dynamic. And to speak the truth, for at least in Italy and France, is no longer a possibility. We do already have some cases of indigenous dengue and chikungunya. So it's no longer somebody else's disease. It's already our disease also. So it's minds have to change now. And so I really want on this small part to really focus on the role of humans. Because if the disease, if the disease spread a lot in the 18th century, it's because of a lot of exchanges, a lot of business, a lot of support, etc. But that was two centuries ago. Now it's things have changed a lot. We are not doing the same mistakes anymore. It's finished. No, we do a lot worse because we are a lot more efficient now for moving mosquitoes around and for spreading the disease all over the planet. And we are very, very efficient like that, actually. So that's what I was just before going on the concept itself, focus on that point. Another point I would like to focus on, just a snapshot, is another kind of effect, is the impact of stochastic events. Many persons here, since yesterday, mentioned that dengue is very unpredictable. It's very difficult to understand. And it is true. It's very difficult to really understand. It's very complicated. And I have one example about that, is a work we conducted with our colleagues at Pasteur Institute in Cambodia, in Phnom Penh, where we analyzed all the clinical samples and all the samples they had of dengue over 10 years since the national surveillance system has been implemented. And we characterize all the virus, the full length, actually, all of them are full length genomes, and we characterize all the isolate they have, which were circulating. Actually, the 4 serotypes are present. Here you will see only DEV1, DEV2, and DEV3, simply because for a technical reason I cannot really explain, we have never been able to have really a full-length sequence of DEV4. So we were not able to conduct this analysis with the 4th one. We did another kind of work. But what is important is just to mention that we have been able to to identify not the serotype or the genotype. We went even deeper, the different individual lineages that were present in Cambodia over 10 years. And we have been able to follow each lineage independently. So I'm not going into that work, into the history, the phylogeny, etc. The important point is that what we found out, something very surprising for us, is that DEV1, DEV2, and DEV3, for instance, they are totally synchronized in Cambodia. All the independent serotypes and had exactly the same fluctuation of population. In 2003, all the what I would call the old lineages, those present before 2003, all of them have been totally eliminated at the same time. Whatever it was, DV1, 2, or 3. At the very same time, all of them eliminated completely. We could not find them anymore. And so that led to probably a major event in 2003, and they were replaced by what we believe are local lineages, but they could have been imported as well. We just believe they are local lineages simply because they started from a single point in Cambodia, and from this point, over time, they expanded over the whole country. So we suspect it's mostly very local lineages. What we found out also is between 2000 and 2003 in Cambodia there are a lot of major climatic problems, flood, drought, that most likely, but we are at the stage of hypothesis here, but most likely this could, very important climatic disasters or events must have eliminating or deeply affected the circulating mosquito population present. And eliminating the mosquito, they also eliminated the virus. the lineages which were present. And basically, this started over again simply from remaining mosquito pockets, preserved from the different climatic events, and the small pocket that started the process again, and from that spread over the country. And what the question comes then in terms of impact of stochastic events is that who were the survivors? Were not the best, the strongest, the most adapted virus of mosquito? No way. Just the lucky. Just the one, the virus which was present in the mosquito, which by luck was in a small part that was not affected. So we started over with a totally different kind of virus lineage and mosquito. So just to say that anything that any plan we can have, this could be changed very quickly just by stochastic events. And this has been also documented in other places. So when we think about vector control, it's important because we could have a new one that comes with a totally new virus and something different. So, when coming back to dengue quickly now, we know there is no treatment. Vaccine, hopefully yes, and we heard about that today and yesterday. Hopefully yes, as soon as possible, but not yet. It's not ready yet. So basically, the only thing we have to rely on at the moment is vector control. And okay, vector control, good, but not that easy. And we just heard just before, and also vector control doesn't work that much the way it is done. Maybe we have to change our mind and play around with the concepts and think in another way. Mostly it is based on chemicals, by large. And it was also based on eradication, but it's a wrong way. We have the South American example. You eliminate something you know, and you get something you don't know really. It's even worse. It's a basic strategy. It is always better to fight the enemy you know. Know your enemy is the first step. So maybe better not to eliminate, but to change something, the way of seeing the things. And we have additional effect to consider. We now have, and it's still the case, we have permanent movement of populations, mosquito and humans, virus, of everybody. We have to consider the cost. If it is very expensive, especially in time of crisis, to control the disease, maybe people will not do it. We have resistance with chemicals. It's now a very big issue, because many populations are becoming resistant. And if your population locally is not, but another one is coming which is resistant, then you are facing a problem. So resistance is a problem. Acceptance. Would people accept large spray of chemicals with all the problem of health, environmental impact, pollution? No longer. Maybe before is no longer true. And addition to that, even with the chemicals, even if people accept, even if there is no resistance yet, many molecules are now forbidden for use. We have less and less and less and less molecules to be used for mosquito control and pest control altogether. So figure is becoming very complicated, and so we have to consider things in a different way. We have to consider it in a holistic way, in a comprehensive way. We have also to address all the societal issues which are linked to mosquito control and to the disease. It has to be affordable and applicable. So we just said the same thing. And we have, and that's important, we have to make it acceptable. And so I would like to change. No, we are going to change a little bit the words. And what we're talking about mosquito control. Now, the concept we are really talking about is integrated and sustainable management of mosquito control. I prefer a lot this word of management rather than control. Control means I have to eliminate. I build a big wall, a trench, I put a machine gun, and I try to shoot at everything coming. That's control. If I talk about management, it's a lot more subtle, and it's another way of considering things. So personally, I think we have to do management, not control. It's a different way of seeing things. And so we are no longer really a technical issue, whereas we have to get the balance between disease and the benefit of this disease. Rather, we have to move to a comprehensive societal risk-benefit balance. So mostly a societal issue rather than just a technical issue. It's not exactly the same thing. So if I now go deeper into the mosquito control system itself, we have to go away from chemicals, it's obvious. We have to make it safe, sustainable. This translates into biological control. There is no real other possibility. We can always try the mechanical control, but it doesn't work very well. So, so biological control against adults is not easy at all. But we can mimic. We can mimic we can mimic the use of biological control agents against adults by using lethal traps, OV traps, other kind of traps that will attract the mosquito, the adult mosquitoes, especially the female, and not only trap them but kill them also. We can doing that we can some sort of mimic the behavior of a predator. Okay, biological control. For larvae, it's a lot easier because we do have several tools on hand, but it depends on the kind of breeding size, whether it's a small one, a large one, open in a city, etc. It's not the same strategy, it's not the same approach. We can use fish or copepods like Mesocyclops. We can use Wolbachia also. I'm not going to talk about that because there will be a complete presentation on that right after mine. And I'm going to spend a few minutes on that. We can use also another product, which is Bacillus thuringiensis israelensis, or also known as BTI, which is a very successful control agent. What is BTI? BTI is a bacterium. It's called BTI because it was discovered in Israel in the Negev desert in the mid-70s. It's very successful mosquitocidal agent against Aedes, Culex, Anopheles. Also Simulium, so very good against the black flies. And for instance, it's currently the only product authorized in Europe for mosquito larvae control. It's the only one we can use. So it's not only an idea, it's also an obligation, for us at least a legal obligation. So what BTI works well because it's a mixture of different toxins present in the parasite body, we call the crystal here. And two toxins are of the group we call CRY for crystal. The other one important is the site toxins for cytotoxic. So the toxin itself is not a single component, it's a combination of 4 different products, natural products of course. And the good thing about BTI is not only that it kills well the mosquitoes, it also doesn't bring any resistance. And this has been tested for a long time, field, laboratory. There is no resistance to the whole crystal. We can get easily resistance to individual CRY proteins, but as soon as we add the site toxin, we can overcome easily the resistance. So there is no resistance buildup because of the specific role of site, which is does a mediated insertion of toxin directly into the membrane without using the receptor, receptor proteins CRY is using, so it directly binds to the membrane and gets into that. So it kills immediately without receptor, which is very important because there is no protein, no gene associated to this activity. So therefore, no possibility to have a selection from the genetic standpoint, a selection of a given gene for resistance. There is no gene. Direct entry into the membrane, like a detergent. So it's a very successful product with a whole bunch of different formulations, depending on the kind of habitat we want to tackle, and could be developed at very low cost, even on agricultural waste. And the production is done in different countries around the world, and very easy to use actually. Application. Well, it depends also, again, on the kind of habitat. Could be aerial, could be handmade, man-made, hand, could be motorized or not. And there is, we can use a different kind of formulation for that. The only thing which is important is that it requires a very good monitoring. And we come to the last point of the talk. It takes a very important monitoring because the lifetime is quite short, 2 to 3 days maximum. Once in outside. So we come again into the risk management. We cannot put the product and say it will work for a long time. No way. It's very limited, which is a good point and a bad point. And it's also safe for use in drinking water container. Okay. So with that also, this product, which is good, we go also for management. And then I come to the last part of this presentation. I'm going to conclude on that. Oops, I'm very sorry. Okay, which is the need of a real integrated approach. The tools exist. What we need is to have an adapted response. We know that we must have an integrated response and holistic approach, because we're talking about mosquito control, vector control, so it's not the point. What is the real point? What is the real element? What the key factor we have to control? It's just the frequency of infecting bites. We don't care if there are a lot of mosquitoes if they don't bite or if they are not infected. So we go into the management, not into the control. It's different. Okay, it's the frequency of infected bites. So the action is basically to reduce this frequency through the combination of all simple, safe, cheap, and socially acceptable means we can find. That is called integrated management. So for that, it takes a good modeling, a good decision system. We cannot do it at the large scale. It doesn't work. We have to do a system which works at the community level. It's very important. So the point is to be able now to go into a good modeling system. And a system, disease, the spread of a disease is a process. Any kind of process, technical, industrial, administrative, or biological, can be modelized. We can make a model of it. We describe this process. We can understand the different steps. We can understand the different actors. We can put a links between the different actors. This can be really understood and can be modeled. So we just need to apply that to the pathology, to the transmission of the disease. That's what we did, and that's a work I'm going to describe quickly. It's not really on dengue strictly, it's on chikungunya. It's something we did with University of Gadjah Mada this year, actually, with Dr. Trebasco and his group. And but chikungunya and dengue, when it comes to that aspect, is exactly the same thing. A few differences in mathematics, but it's very similar. So we moved from a static model to describe, and from that we developed software, a computer program for simulation, modeling and simulation, which we applied to real conditions in Yogyakarta and in Sukoharjo. And and this is important because we have been able to test some scenarios. But again, I insist, if you want this to work, we have to address it at the community level, not at a very large level. It is too complicated otherwise. We cannot. And for instance, just to show, no need to go into the details, this is the complete class diagram of the chikungunya and dengue pathosystem. It's the same for both. Where all the actors, all the links, all the interactions are described, whether it is the sample themselves, the clinical aspects, the biology, which we mean by biology is the genetic diversity, the description of the different populations we have, or lineage in terms of virus, also all the geography, environment, anthropic action, or ecology, like the weather, temperature, humidity, geography change of habitat, etc. Everything can be integrated in the same model if we use a multi-agent system. Of course, this is also must lead to a real program. Therefore, it is also considered into a different system. With it's not the multi-agent; it's the partial equation, differential equation system. But here's the mathematics for chikungunya. For dengue is different, but only the mathematics is different. It's a bit more complicated for dengue because we have the vertical transmission as well. So what I just want to show quickly is that this is something we applied here. When here in Yogyakarta, and next to here in Sukoharjo with Dr. Trebasco group is here, and we worked together on that project. And we have been able to collect all the data in one RT in Sukoharjo, RT number 3, rural area, collect all the data, geographic, the population, the exact location of the houses, all the epidemiological and clinical data, who was sick, who was the first case, the second, how long, what time, etc. We got all this data, and we have been able to integrate that into a simulated RT. in the program itself, with the exact number of people, the exact number of houses, the good distance, etc. And then it works also on 3D, and also to recreate on computer, recreate online the actual outbreak, how it worked, what happened. We found out that, for instance, for the outbreak to start the way it started in Socorro, we need at least 2% of infected mosquitoes. We tested different elements, like introduction of affected human on infected mosquitoes. And we did the same simulation in a peri-urban area in Yogyakarta, with exactly the same things. And there we have been able to identify different elements which were very important for the spread of the disease, or in contrary, very important for reducing the spread of the disease, diffusion of the disease. And we tested several scenarios, of course, with the idea of reducing the frequency of infecting bites, which comes to a combination of action, very simple. And so we found out, and I'm concluding on that, we found out that basically in this case, for chikungunya, will not be the same conclusion for dengue. I'm talking about chik. The conclusion might be different. The three main factors is the regular introduction of mosquitoes, infected mosquitoes and or humans, exogenous in the village, amplification by the local mosquito population, and the persistence of outbreaks due to the health system where the viremic people are sent back to the village. And they can be a starting point for secondary infection in the village itself. And so this is kind of, we say, a community-based approach that we can apply from now. This will not work at a very large scale. It's not possible to integrate everything. But if we switch from one single large system that does not work to a whole series of smaller actions, community level, with a good planning, then it could work. And with this, I will thank you for your attention and finish this talk. Thank you very much. Terima kasih.